Hello students, good afternoon to everyone. I hope you all are doing great. So yeah, already in the morning we have solved sample paper 1. Now we are here with solving sample paper 2. Right? So before starting the session, let me introduce myself. This is your biology master teacher Navya, and
This is your chemistry master teacher Suhas.
I hope you all had your lunch, right? Okay, what is this, sir?
Sorry, ma'am.
Okay, fine. It's okay. So I hope you all would have had your lunch, right? I know you all might be feeling a bit drowsy right now. It's already it's already 2:40 right
46.
now.
Yeah,
Yeah, no, they might be feeling drowsy. So let's try our level best to make the session very interesting, right?
With fun.
Yeah. So now I want you all also to answer to this question. Only after that, I will explain you the concepts. Try to answer these questions as well now. Okay?
Yeah.
So anything you
Yeah,
want to
ma'am.
tell, sir?
No, ma'am. After Navya ma'am, I came. Don't worry. And after
Yeah.
Neha ma'am is coming.
Yes.
Thank you, ma'am.
So we'll be starting with
Bye, bye, students.
biology now. So yeah, let me. Okay, so am I audible, students? Am I audible? Is the PPT everything clear? Let me check once. Yeah. Okay. So now we are good to start. Right? So first we will solve biology questions. Right? Okay, in between if you have any doubts, feel free to comment in the comment section and I'll clarify your doubts. Okay? Volume is low. Is it? Let me check. Right. Okay. No, pro learner, it's fine only. Just check once with your device. Okay. So yeah, let's start with our first question. Okay. So the first question says that amoeba shows following kind of nutrition. Right. So which means to say this question is picked up from life process chapter. Right. Yeah. Okay. So voice is clear. Okay. Thank you for confirming. Okay. So yeah, amoeba shows following kind of nutrition. Option A says autotrophic, option B says holozoic, and option C says saprophytic, and option D says parasitic. Okay. So you say that it is holozoic, right? So S, option B is the right answer. It is holozoic mode of nutrition. So while doing life process chapter, already we started the first topic that was there. Which was the first topic we started in life process chapter? It was nutrition, right? Wherein we studied autotrophic mode of nutrition as well as heterotrophic mode of nutrition, right? So this holozoic mode of nutrition, it falls under which type of, which mode of nutrition? It is, it is heterotrophic mode of nutrition. It's a type of heterotrophic mode of nutrition, right? What was happening in autotrophs? See, those living organisms which prepare their own food, they fall under autotrophs. Whereas on the other hand, those living organisms which are dependent on another life form for their food, those living organisms are considered as heterotrophs and their mode of nutrition is called as heterotrophic mode of nutrition. Right, you are clear with that. Now, and then again under heterotrophs, there were three types. One was holozoic mode of nutrition, and then there was parasitic mode of nutrition, and then there was saprophytic. So now you understood that yes, amoeba falls under holozoic mode of nutrition. Why do you think so? Why it is called as holozoic mode of nutrition? What was happening? See here, what is amoeba doing first? See, whenever there is food particle near amoeba, first the amoeba, it will, what is it doing? It will enhance its finger-like structure, like what is this called as? It is pseudopodia, and that pseudopodia, it will engulf the food particle and it forms food vacuole, right? And then So this is called as egestion, and then once the food vacuole is formed, then what happens? The food within the food vacuole, it is digested. How? Because the food vacuole, it releases certain digestive enzyme, and those digestive enzyme will digest the food particle that is present in the food vacuole. So that is digestion step. And then what happens? After digestion, the next one is absorption. The digested food is now getting absorbed within the cell. Right, it is getting distributed evenly within the cell wherever it is needed, and that is called as assimilation. So after digestion, absorption and assimilation is taking place, and after that there will be some food material, right? There will be some food material which is not needed for the amoeba, right? And even it won't have been digested properly, maybe because it is waste material for amoeba, right? Such materials will be excreted out, and that step is called as egestion. And this is Holozoic mode of nutrition. What was happening in saprophytic mode of nutrition? Over there we saw, right, mostly fungi and all that. What do? They derive their nutrients from dead matters. That's why they were saprophytes, and that mode of nutrition is called a saprophytic mode of nutrition, right? And what about parasitic mode of nutrition? Over there, the living organism, what were they doing? They were either staying on the host, or they used to infect the host, and from that host they were deriving nutrients. That's why that is called as parasitic mode of nutrition. And in our question they have asked about amoeba. So it is Holozoic mode of nutrition, that is the answer. Okay, clear with this? And now I want you all to think. See, you all would have heard about insectivorous plant, right? Yeah, no, you know, right, insectivorous plant. See, I already told you plants, they are autotrophs, which means to say they prepare their own food. Now, what about insectivorous plant? Insectivorous plant like Venus trap and all that, why do they capture the insect? What? Pro-learner says living organism take food without killing the organism. See, now, I told you, insectivorous plant, what do you think about insectivorous plant? See, you all know that they trap insect, right? So now, do you think they fall under autotrophs or heterotrophs? I am talking about insectivorous plant. Because see, insectivorous plant, based on this concept, 2-mark question or 3-mark question can be asked in your board exam. Because nowadays they prefer asking competency based question also. So anyway, I am doing nutrition part, right? So I thought, okay, let me explain about insectivorous plant also, I thought. So yeah, they contain digestive enzyme inside them. No students, I want you all to guess if the insectivorous plant, whether it is autotrophic or heterotrophic. Because insectivorous plant, they trap insect, no. Why are they trapping insect? So it's because they are trapping insect. Do you consider them as autotrophs or heterotrophs? Any idea? Any guesses? If not, I will give you the explanation now. So if anyone knows about that, then you feel free to comment in the comment section. Any guesses? Okay, so let me explain. See, insectivorous plants, see, they are green in color, which means to say that they have chlorophyll pigment. So this insectivorous plant, they perform photosynthesis, which means to say that, no, actually, see, I am telling you now, insectivorous plants, they perform photosynthesis, okay, they obtain their food, they prepare their food through photosynthesis process, okay. But then see, what are they preparing after photosynthesis? What will plant generate? They prepare their food, that is their carbohydrate. They get glucose, right? They get their glucose. But then, now, okay, forget about plant. What about us? What is present in our food? Usually our food will have carbohydrate, protein, fats and all that, right? Along with that, we even want vitamins and minerals for our overall growth and development, right? So similarly, plants also, along with carbohydrate like glucose, the plant also require certain minerals. Okay, certain minerals. I also told you, for photosynthesis, what is the raw material? It is water, right? Along with water, the plant even absorb mineral from the soil, right? Which means to say that plant require carbohydrates and other minerals also. There are certain macronutrients and micronutrients which plant do require for their overall growth and development. Okay, so what happens in case of insectivorous plant is that, see, the insectivorous plant, they prepare their food via photosynthesis, wherein they get their carbohydrate. But then these insectivorous plant, they don't get nitrogen. They don't get nitrogen. Usually those insectivorous plants, they are grown in a soil where the nitrogen content is very less. But then plant require nitrogen. Only then they are able to provide good fruits. They want for their overall development, right? That's why these insectivorous plant, it's because they are grown in a soil where there is no much minerals, where there is no much minerals. That's why those insectivorous plant, they will trap the insect, and from that insect they will derive those nutrients which they are not getting from the soil. Okay, so insectivorous plants are such type of plant which perform both autotrophs and heterotrophs. Keep this in mind. Okay, insectivorous plant, they are autotrophs also, and they behave like heterotrophs also. Why? Because they trap insect. Why are they trapping insect? In order to derive the nutrients, certain minerals like nitrogen. Okay, clear with that? Okay, so now let's see our second question. So you understood the concept, right? What is happening in insectivorous plants? All clear? So if you understood the concept, do send me a thumbs up in the comment section. Okay, so now let's move on to question number 2. So here the process by which the blood is cleared of metabolic waste in case of kidney failure is called as. It is called as. Option A says artificial kidney. Option B says dialysis. Option C says transplantation. Option D says filtration. Okay, so you all understood the concept. Okay, so now which one do you think? Anurag, right? Anurag says option A, but Anurag, option A is not the right answer. Yes, it is option B. Option B is the right answer. See, even in your NCERT textbook, you would have seen, if you want, you can open your NCERT textbook now itself. You would have seen after that excretion part, there is one table. Within a table, there is a content which is explaining about, there is a paragraph which is explaining about dialysis. Okay, so let's see what is happening in dialysis. See here, the process by which the blood is cleared of metabolic wastes. Metabolic waste like nitrogenous waste like urea, uric acid, ammonia and all that. How is it getting cleared from our blood when the kidney is not working properly? You should keep this in mind. When the kidney is not working properly. So they have mentioned clearly in the question that when the kidney is not working properly, then how is blood purified? Right. So what is the function of kidney? Kidney, it is involved in the formation of urine. Right. How is urine being formed? First, the blood, it carries nitrogenous waste from liver to the kidney, and within kidney, there are certain filtration units. Those filtration units are called as nephrons. Right. So those nephrons, they used to, what was the function of nephron? They used to filter the blood. They used to filter the blood from which the nitrogen waste were removed, and then urine was formed. Right. So, but then when the kidney is not functioning properly, it means to say that nitrogenous waste cannot be removed. So that time if the nitrogenous waste, if it gets accumulated in our body, then it can be toxic to our body. It might even it might lead to death also. So therefore it is very important to remove those nitrogenous waste. Okay, so that time they will perform dialysis. Dialysis looks somewhat like this. So what are they doing in dialysis is that, see, from the person's body, the blood is taken, and then in the dialyzer what happens is that the blood is purified. The blood is purified, and then here the nitrogenous waste is removed from the blood, and then the purified blood will be circulated back into the patient. Usually kidney failure patients, for them they carry out this dialysis. That's what they are doing in dialysis. Okay, because their kidney is not functioning properly. And now I want you all to think that from where is this nitrogenous waste being produced? From where? See, you know how, from where our body, see, you understood that how carbon dioxide was produced. It was after respiration, the carbon dioxide was released as a byproduct, and then the blood used to carry that carbon dioxide, and then the deoxygenated blood, when it reaches the lungs, over there the carbon dioxide was diffusing into the alveoli. From alveoli we were exhaling that carbon dioxide out. Okay, fine, you understood from where the carbon dioxide came. It was during that respiration process. Okay, fine. Now what about nitrogenous waste? From where did nitrogenous waste come from? It is, see, from proteins only. After the breakdown of protein or after the death of protein, or there are certain reaction in which if the protein is involved, the byproduct will be released as urea, uric acid or ammonia. Okay, they are poisonous substance which should not be, it should not get accumulated in our body. Okay, so those nitrogenous waste is being removed. Okay, clear with that? So let's move on to the next question. So now, during digestion waste, yes, from there also certain, nitrogenous waste will be released. Okay, clear with that? So we'll move on to the next question, that is, where does digestion of the starch begin in human body? Where does digestion of starch begin in human body, students? Any guesses? Class 10, hello, hi. Yeah, so where does digestion, yes, it is mouth. Nothing but mouth is also called as buccal cavity, right? So you all have already studied. So what is present in mouth which is causing the digestion of starch? What is present in buccal cavity? What is being secreted in buccal cavity? Hello, hi everyone, hi. Yeah, so what is happening in mouth? What is happening in buccal cavity? How is that starch being digested in buccal cavity? And do you think that entire carbohydrate digestion, is it taking place only in buccal cavity or does it take place even in small intestine? Any guesses, students? Yes, it is converted into sugar. Maltose. Yes, starch will be broken down into maltose. By the action of which enzyme? By the action of which enzyme? It is salivary amylase. Right. So the first thing is the buccal, what is present in saliva? Saliva is a digestive fluid which carries enzyme, digestive enzyme which is called as salivary amylase. Okay, good. So you all are answering very nicely. Yes, good. Yeah. So first, when we eat food, see, in our buccal cavity, there are two types of breakdown is happening. One is mechanical breakdown of food and the second one is enzymatic. How is mechanical breakdown taking place? See, in our mouth there is teeth, right? First you chew the food nicely, that time the particles will be broken down into tinier ones, right? After that what happens? That is the mechanical breakdown. And then what's happening? Then the saliva will get mixed with the food, right? What is present in saliva? It is salivary amylase. So this salivary amylase, it breaks down the starch that is present in our food into maltose. So in buccal cavity, partial digestion of carbohydrate is taking place. Okay, again maltose will have to be broken down. See, this is not the complete digestion. Here partial digestion of carbohydrate is taking place. Again the complete digestion takes place in small intestine. Okay, clear with that? And then in our mouth, in our buccal cavity, how many salivary glands are there? It is 3. 1, 2, 3. See, again in MCQ question they might ask, how many salivary glands are present in our human body? It is 3. Okay, clear with that? Fine, this is all about your digestion of starch in buccal cavity. Clear, students? Shall we move on to the next question? Fine. The question says, during contraction of heart, what prevents backflow of blood? What prevents backflow of blood? Option A says thin walls of artery. thick muscular walls of ventricles, walls in heart, and all of the above. Which one do you think is the right answer, students? During contraction of heart, what prevents backflow of blood? Which one do you think is the right answer? Come on, student, answer. What's the answer for this question? What is preventing backflow? Is it the chamber that is present in the heart, or is it the walls that are present, or is it all of the above? It is? What's the right answer? I want you all to answer to this question. I know it's 3:10 right now, you all might be feeling drowsy, but then let's make it interesting by making this session interactive. Right, so I want you all to answer. Why during contraction of heart, what prevents the backflow of blood? Okay, so it is option C, yes. Walls that are present in heart that prevents the backflow of blood. Yeah, so the thing is, there are how many walls are there in our heart? There are 4 walls. Okay, you must even know the names of these walls that are present in the heart. Okay, so you know this, right? This is your left atrium and this is left ventricle, and this is right atrium and this is right ventricle, right? And now that is your aorta, right? This is your aorta, and then this is your pulmonary, that is pulmonary, the one that is going out, that is your pulmonary artery, and then there is one more wall that is coming in, that is pulmonary vein is also there. Okay, so now the valve that is present between left atrium and left ventricle, that is called as bicuspid valve. This is called as, that is named as bicuspid valve. Okay. Yes, and then the valve that is present between right atrium and right ventricle, this is tricuspid valve, right? And then the valve that is present between the left ventricle and this is aorta, right? Left ventricle and aorta, that is your aortic semilunar valve. That is aortic semilunar valve. And then the valve that is present between, there is one more valve that is present over here between right ventricle and that is pulmonary artery which is carrying the deoxygenated blood from the heart to the lungs. So the valve that is present between the right ventricle and the pulmonary artery, that is called as pulmonary semilunar valve. That is pulmonary semilunar valve. It would be better if you try to remember these names, valve, okay? Because see, even if you are preparing for your NEET exam, it will be useful. That's why you must know what are all the types of valve that is present in the heart. Okay, so the valve, you know that it is preventing the backflow of the blood. Okay, along with that you must learn the types of valve that is present between the artery and ventricle and the artery and the veins, the blood vessels. Okay, clear with that? So, shall we move on to the next question? So, the next question is differentiate between single and double circulation that is found in vertebrates. Why is it called as double circulation? Can anyone say in just one sentence that why the blood circulation in human being, it is called as double circulation? I want the answer in just one sentence. It's very simple. Right, see, you will have to differentiate. This can be asked for 3 mark in your boards. Bicuspid wall is also called as mitral wall. It is also called as mitral valve. Okay, so what happens is that, why is it called as double circulation? It's because the blood, it flows through heart twice. Initially as it initially in the deoxygenated form and then oxygenated form. That's why it's called as double circulation. Whereas you see single circulation in aquatic animals like fishes. Over there what happens is that first once after the oxygenated blood, after it delivers the oxygen to the cells in the body, then what happens is that the deoxygenated blood, it will flow towards heart, and then the heart, it will pump the deoxygenated blood to the gills, wherein in gills what happens is that the deoxygenated blood will be converted to oxygenated one, and then that oxygenated blood directly from the gills, it is being supplied to the other parts of the body. That's why in aquatic animals like fishes, you get to see single circulation, wherein only once the blood flows through heart, that too in deoxygenated form. Yeah, feel free to comment in the comment section if you have any doubts. Okay, clear? Fine. And then in case of double circulation, what happens is that you can see in this image, see first from different parts of the body, the veins carries deoxygenated blood. It carries deoxygenated blood from different parts of the body to the heart, and then from heart the deoxygenated blood is pumped to lungs. Which blood vessel carries deoxygenated blood from heart to the lungs? It is pulmonary artery. Pulmonary artery carries deoxygenated blood from heart to the lungs. So in lungs what is happening? The deoxygenated blood will be converted to oxygenated blood. After completing biology, chemistry will be started. Okay, somewhere around 3:45 4, chemistry will be the Suhas sir will be taking chemistry. Okay, yeah. So what happens then in lungs? The deoxygenated blood will be converted to oxygenated one, and then the oxygenated blood will be carried by pulmonary veins to the heart, and then from heart the oxygenated blood will be pumped to the different parts of the body. So you understood, right, what is single circulation and double circulation. So now they are asking you to differentiate. So here see single circulation, what is happening? Like I told you, the blood only once it passes through the heart, that too in deoxygenated form. Okay, and then double circulation, what happens? It passes twice, once in oxygenated form and the second time it is deoxygenated form. Okay, and then heart has only here, that's what deoxygenated form. Over here it is oxygenated and deoxygenated form, and here the efficiency is very less. In single circulation the efficiency is very less, whereas in case of double circulation the efficiency is more. Okay, this question it can be asked for 3 mark, and trust me students, double circulation is a very important topic. It can be asked for 5 mark also. Double circulation, be thorough with the topic. Every year from circulation part there are questions, from transportation system, that to human circulatory system, there are questions that is being asked directly for 5 mark. Okay, fine. So let's see next question. What is the difference between artery and veins? So yeah, like you all know, see artery, always it carries blood away from the heart. It carries blood away from the heart. Whereas vein, it carries blood from different part of the body towards the heart. Always remember that artery away. It carries blood away from the heart. And then veins, it towards the heart. Right. So it's because see the artery, it carries oxygenated blood from heart to different part of the body, right? So when the heart pumps that oxygenated blood, the pressure is very high. Therefore, to tolerate the pressure, the artery's walls are very thick. The walls of the artery is very thick when you compare with veins. Whereas when it comes to veins, see here, what is it doing? It is carrying the deoxygenated blood from different part of the body towards the heart, right? So when it is carrying blood from different parts of the body towards the heart, the pressure is less. Therefore, the wall is bit thin. Therefore, the wall is bit thin when you compare the arteries with vein. Veins have thin walls, whereas artery have thicker walls. They have thin walls. But then, in order to prevent the backflow of the blood, blood in veins, there are valves that are present. Veins, they also have valves. Okay. Valves are present in veins, whereas in artery there is no valve. Okay, clear? Fine. And also they have the lumen is narrow. The lumen is narrow. Students, if you have any doubts, feel free to comment in comment section, right? And in case if you have doubts more than 2-3 questions, then write them down at the end of this session. Like after completing biology question, I will allot 5 minutes time for you all so that you can ask questions and I will clarify your doubts. Okay? Clear? First we'll solve these questions. Now let's stay focused. We'll focus on these questions because these are very important questions from life process chapter. That to see majority of the question will be asked from life processes every year. Okay, so first we'll finish these questions and then I will solve all your doubts. Okay, clear with that? Shall we focus on this now? Yes. Gagan, you have doubt in some question? Which question, Gagan? Previous one? In the previous one you have doubt, Gagan? Okay. So like I was telling, here the walls are present only in veins and not in artery. Okay. And then the lumen is wide when it comes to veins. The space is more, whereas in case of artery, the lumen is less. Obviously because the wall is very thick. Okay. And then here I told you the pressure will be very high because the oxygenated blood is being pumped by the heart with high pressure. That's the reason. Okay, whereas when it comes to vein, the pressure is low because the deoxygenated blood is being carried from different part of the body towards the heart. That is the reason. Okay, now I told you artery carry oxygenated blood and the veins carry deoxygenated blood. Okay, whereas the pulmonary veins, you must remember this. Pulmonary veins, what do? They carry oxygenated blood from heart towards lungs. From lungs towards heart. Which one? Pulmonary veins. Whereas pulmonary artery carry deoxygenated blood from heart to lungs. Okay, artery carry oxygenated blood from heart to different parts of the body, whereas pulmonary artery carries deoxygenated blood from heart to the lungs. Okay, and then whereas veins carry deoxygenated blood from different parts of the body towards the heart, whereas pulmonary veins they carry oxygenated blood from lungs to the heart. Okay, most of the students they get confused with veins and pulmonary veins, artery and pulmonary artery. So don't get confused, just note down this. Okay, always artery they carry blood away from the heart. They are carrying away. So here artery it is carrying oxygenated blood away from the heart. Pulmonary artery is also carrying deoxygenated blood away from the heart. Clear with that? Okay. Fine. So let's move on to next question. What is villi? What is villi and what are its function? What are its function. So here, you know, where is villi present? Where is villi present? It is present in small intestine, right? So now what is happening in, so before getting into the concept villi and what is its function, first of all, what is happening in, hi master quiz, hi everyone. So yeah, what is happening in small intestine? We have already studied, right? In small intestine, the complete digestion takes place. The complete digestion of food takes place. After that, there is absorption that is taking place. Why absorption? It's because what is present in small intestine. See, if you zoom in, that is present, right? So now, what is present within the villi? There is blood capillary that is present. Yes, not just complete digestion of fat, complete digestion of protein, carbohydrate, fat, everything takes place in small intestine. See, I told you, in buccal cavity, the starch is broken down into maltose, right? Starch is broken down into maltose. Okay, Gagan, you have doubt in fish wala thing. Yeah, what is your doubt? Oh, okay, in that single circulation you have a doubt, Gagan. You have doubt in single circulation? So, where I was? Yeah, I was telling you that in buccal cavity the starch is broken down to maltose, right? That's it. Again, maltose will have to be broken down into the simplest form, that is glucose. That breakdown of maltose to glucose takes place in small intestine. And also protein, where is protein breakdown taking place? In the stomach. Okay, the proteins are broken down to peptones. Again, peptones will have to be broken down. It has to be broken down into amino acids. Yes, so I guess students, you have doubt in single circulation. Ma'am, single circulation, it happens in fish. Yes. Yes, it's because see, the heart, they have only one chamber, they have only two chambers. Yeah. So yeah, where was I? Yeah, in stomach the protein, it is broken down to peptones. Okay. Again, the peptones will have to be broken down to amino acids. So there is already the partial digestion of protein and partial digestion of carbohydrate has taken place. But then the fat digestion takes place only after entering into small intestine. Okay, so in small intestine, the partially digested protein, the partially digested carbohydrate and the fat will be broken down completely in the small intestine by the action of pancreatic juice, even bile juice, as well as intestinal fluid. Okay, as well as intestinal fluid. Complete digestion takes place. After complete digestion, what happens? There is villi. Now this villi, the inner lining of the small intestine has got so many finger-like projection. See, ma'am, gills meaning, see in our body we have lungs, right? We have lungs, no. So there is a student who is asking about gills, right? See, yeah. Okay, Gagan also has a doubt in single circulation, right? Okay, let me move on to that slide now. Yeah. See, gills, it is nothing but see how in we human beings we have lungs. We human beings have lungs from which they are taken, oxygenated and exchanged with the oxygenated blood. Yes. See here, gill is nothing but, see it is like lungs. How we human beings we have lungs. Similarly, fishes they have gills. Okay. That is their respiratory organ. Okay. Clear? Yes. From which they are taken, oxygenated. Yeah, it is right. Your explanation is right, dear. That is right. See, what is happening is that in single circulation, the blood flows only once through the heart. See, this is the heart. Okay. You can see half of the blood vessel, it is red. It is represented in red color. Is this diagram clear, students? Is this diagram visible for you all? See, initially, the first half, it is red in color and the other half, it is blue in color, which is indicating that, see first what happens, the blood, and this is the gills, right? From gills, the deoxygenated blood is converted to oxygenated one. It is converted to oxygenated, right? That oxygenated blood is circulated throughout the body. Okay. And then the oxygen is utilized by the cells. Why? For respiration process to take place. Why? Because that they require energy, that's why. Right? The oxygen will be delivered. After respiration, carbon dioxide is released. That carbon dioxide is carried by the blood only. So in that blood, oxygen will not be there, instead carbon dioxide will be there. So it is a deoxygenated blood. So that deoxygenated blood, again it reaches the heart. Okay, it reaches heart. In heart, what is happening? The heart will circulate, again it will send the deoxygenated blood to the gills. So like this the circulation is taking place. Understood, Gagan? Okay dear, you are welcome. Gagan, was this your doubt that what is happening in single circulation? Understood? Clear with that? So there is only once, the blood flows through heart only once, that too. Wait, let me show you. Yeah, 1 second. Is this clear? See, it flows only once. It flows only once through heart. The two deoxygenated form. Any doubts? Yeah, was talking about villi, right? So in small intestine, the complete digestion takes place of carbohydrate, proteins as well as fat. Okay, after that it is being absorbed in the small intestine only because of the presence of villi. What are these villi? Villi, it is the finger-like projection which is present in small intestine. Again within villi, like you can see, there is blood capillary that is present. Okay, so the villi, what is the function of villi? This villi, it increases the surface area for the absorption of digested food. Okay, so it increases the surface area and food is getting what? The digested nutrients, it will get diffused into the blood capillary. In blood capillary, the blood is present and that blood will carry those nutrients to the required cell in our body. Right? So the question was, what is villi? It is the finger-like projection which is present in the inner lining of the small intestine. Right? And what are they doing? They are increasing the surface area of absorption. Okay? So this can be asked for 1 mark or 2 mark. Okay? Clear with this? Fine. So let's move on to next question. What type of respiration takes place in human muscles during vigorous exercise and why? So which type of respiration takes place in the muscle cells? Comment in the comment section which type of respiration is that which is taking place in the muscle cell. Is it aerobic or anaerobic? Is it aerobic or anaerobic? And that respiration also has got another name. So can you type in the comment section, student, what is that which type of respiration is taking place in the muscle cell? Aerobic? No, in muscle cell. In muscle cell it is anaerobic. Yes, it is anaerobic respiration that is taking place in muscle cell. Like you all know, see, during vigorous workout what happens? See, your muscle cells, they will have to continuously contract and relax. Only if the muscle cells contract and relax, only then the movement is possible. Right? So if the cells will have to contract and relax so fastly, then they will require energy. Right? So what is happening in muscle cell is that the glucose is broken down to release energy in the absence of oxygen. They don't require oxygen. First the glucose is broken down into pyruvate, and then in the absence of oxygen the pyruvate is broken down to release energy, that is ATP. How many molecules of ATP are released in aerobic respiration and how many molecules of ATP are released in anaerobic respiration, students? How many molecules of ATP is released in aerobic respiration and how many molecules of ATP are released in anaerobic respiration? Can you all tell how many? Right. So in the muscle cells where the anaerobic respiration takes place, along with ATP, even lactic acid is also released as a byproduct. Lactic acid as well as carbon dioxide is also released as a byproduct. So when the lactic acid gets accumulated in the muscle cell, Gagan says 6. Are you sure with that, Gagan? Do you think it is 6? In anaerobic respiration, is it 6? Okay, so what happens is that this lactic acid will get accumulated. It will get accumulated in the muscle cells, and then that will lead to cramp. That will lead to muscle cramp. Okay. So during vigorous exercise, anaerobic respiration takes place, and then what happens is that along with ATP, even lactic acid is also released. Once the lactic acid gets accumulated, that will lead to muscle cramp. That's why when you run too much, when you do too much of exercise, you get muscle cramp. Why? Over there anaerobic respiration will be taking place and the lactic acid will get accumulated. Okay, fine. So actually in anaerobic respiration 2 ATP molecules are released, whereas in aerobic respiration 36 molecules are released. Okay, clear? Fine. Gaurav says, you want me to speak? First we'll do one thing, Gaurav. We'll finish off all the questions, okay? We'll finish off all the questions and then we'll speak, right? Because see, your exams are nearing, boards are nearing. It's very important, right? Now we will have to learn and understand the concept, and also presenting your answer is also very, very important, right? For which, what? Answering the question is very important, right? So yeah, the question was all about which type of respiration takes place in muscle cells, right? Okay. So now see the respiration in human beings. What happens is that aerobic respiration also takes place. See, in higher organism, aerobic respiration takes place, and in the lower organism, anaerobic respiration takes place. So now let's see about all types of respiration. Okay, so there is aerobic as well as anaerobic, because question can be asked from this also. Say if it is aerobic respiration, you get to see them in plants and human beings also. Okay, so over here what is happening? In case of aerobic respiration, first the glucose will be broken down into pyruvate in the cytoplasm. Okay, and in case of aerobic respiration, what happens is that the pyruvate will have to be broken down to release ATP in the presence of oxygen, That's why the pyruvate enters into mitochondria, and in mitochondria the oxygen will be available. Over there the pyruvate will be broken down in the presence of oxygen, and then energy is released, that is ATP is released. That too 36 molecule of ATP will be released. Along with that, carbon dioxide and water molecule is also released. Okay, clear? So the location of aerobic respiration is initially the breakdown of glucose. The breakdown of glucose takes place in cytoplasm as well as mitochondria, right? And then in case of anaerobic respiration, so this is our anaerobic respiration. Anaerobic respiration, it takes place in muscle cell also, and you get to see anaerobic respiration in some of the yeasts as well as this one. What? Yeast cell as well as certain bacteria as well. Yes, Gagan, majority of the questions are from life process only. In this sample paper, most of the questions were being asked from the life process chapter. Okay, fine. So yeah, here what happens is that the anaerobic respiration, it completely takes place in cytoplasm only. Okay, so here the glucose is broken down into pyruvate, and then the pyruvate will be broken down to release, in case of yeast cell, ethanol is released, carbon dioxide and also energy is released, and 2 molecule of ATP is released. 2 molecules of ATP is released. Okay, same thing lactic, we saw in muscle cell how it is taking place. Over there, along with ATP, lactic acid is also released. These two are anaerobic respiration, whereas this is aerobic respiration. Clear with respiration part? See, respiration topic is also very important. From this, most of the questions can be asked. Okay, any doubts in the respiration part, students? Let's move on to next question. Yeah, actually, you know what? Yeast, no, they see this. The anaerobic respiration that is taking place in yeast, it is also called as alcoholic fermentation. Actually, it is used in industries. The yeasts are used in industry. in preparing ethanol, alcohol and all that. Okay, fine. So we'll move on to next question. The difference between aerobic and anaerobic respiration. I just explained you, right? So again, this can be asked for 3 mark or 5 mark. Fine. See, aerobic respiration, it takes place in the presence of oxygen, whereas anaerobic respiration in the absence of oxygen, right? Okay. And then it occurs in cytoplasm. The location is aerobic respiration, it occurs in cytoplasm as well as mitochondria. Whereas anaerobic, it occurs in cytoplasm only, like I showed you. See, I have shown you. Fine. And then complete breakdown of glucose takes place, whereas over here incomplete breakdown of glucose takes place in case of anaerobic respiration. That's why you are getting only 2 ATP molecule, whereas in case of aerobic respiration you get 36 molecule of ATP. Why? Because complete breakdown of glucose is taking place in case of aerobic respiration. But then in case of anaerobic, it is incomplete breakdown of glucose. That's why you are getting only 2 molecule of ATP. Okay, clear? And then end products are carbon dioxide and water, whereas in case of anaerobic respiration, the end products are carbon dioxide, ethyl alcohol or lactic acid as well. Okay, fine. So next question, what differentiate between the transport of material in xylem and phloem? So it can be asked for 2 mark, right? So here what is happening? See, they are asking about what type of food material is being transported in xylem and phloem. See, xylem, you all know, it will transport water and mineral, right? Whereas phloem, what is phloem doing? It transports food material from leaf to different parts of the body. Okay, please students, I will explain all, you write down your questions in the comment section, I will get back to you. Now let me finish off these questions and then I will clarify your doubts. Okay, fine. So here, and then always the movement in phloem is bidirectional. Just a second. In phloem it is always bidirectional, and then in xylem it is unidirectional. In xylem it is unidirectional, whereas in phloem it is both upward and downward, it is bidirectional. Okay students, clear with that? So we'll move on to next question. What is the role of glomerulus in kidney? What is the role of glomerulus in the kidney? I already explained you in the dialysis part. In the dialysis part, I already told you that nephrons, they are the filtration unit in kidney. They filter the nitrogenous waste from the blood and the urine is formed, right? So now what is this? See, this is how the nephron look like. This is the structure of nephron. See, this part is present here. What is this? See here, the blood capillary that you can see, that is glomerulus. That is glomerulus. And then this capsule kind of stuff which you can see, that is Bowman capsule. That is Bowman capsule. Okay, from Bowman capsule you can see that there is a tube-like structure. There is a tube-like structure that is coming out. There is a tube-like structure that is coming out from the Bowman capsule, and then this long tube that will lead to collecting duct. Right. So what is your glomerulus doing? So first what happens is that, let me tell you, first what happens is that the blood, it from the efferent arteriole, this is efferent arteriole and this is afferent arteriole and this is efferent arteriole. First the blood, it enters through the efferent arteriole to the glomerulus. In glomerulus, when the blood flows through these tubes, the water, what is it? The waste material is filtered from the blood. Along with waste material, certain important substances like amino acid, glucose are also filtered. They are also filtered. Am I not audible, students? Ravi, am I not audible? Is my voice clear? Okay, am I audible, students, or is my voice not clear? Let me check once. Yes, students, I am audible, right? Okay. Yes, I guess it is audible, students. Okay, fine. Where was I? So what happens is that the entire here the blood, when the blood carrying nitrogenous waste, when it flows through this glomerulus, here what happens is that along with the nitrogenous waste, the important elements are also important substance like amino acid, glucose, everything will be filtered. Okay, that will get collected in the Bowman's capsule, and then the blood comes out through efferent arteriole. It comes out through efferent arteriole, and then this efferent arteriole will join to this blood capillary which is surrounding the tubule. Okay, and then in Bowman capsule, along with nitrogenous waste, some certain important topics are also, important substances are also there, right? When that fluid flows through this tubular structure, the blood capillary, it will absorb the important substance from that fluid. Okay, clear with that? Fine. So here glomerulus, what is it? It is actually a capillary that is present in Bowman's capsule. It will receive the blood from renal artery which will carry the waste, nitrogenous wastes, right? And then it filters the waste material. Along with that, certain important substances are also, certain important substance also will be absorbed, right? And then it will be containing end product of protein and yellow bile compounds from the liver as well. That's what the glomerulus is doing. It is just filtering out. Along with waste material, it is also filtering out the even the important substances as well. And again, that important substances will be reabsorbed back when that fluid passes through the tubules. Okay, and then the waste fluid, that is the liquid which is carrying nitrogenous waste, that reaches here, and that will lead to the collecting duct wherein the urine will be collected, and then it comes out of the kidney through ureter, and then it will be collected in the urinary bladder. Okay, clear with this? Okay, fine. So next question, what is the advantage of fourth chamber of heart? It is very simple. So over here what happens is that the oxygenated blood will not be, in order to prevent the mixing of oxygenated blood with the deoxygenated one, to prevent the mixing, there is fourth chamber. Why? Why should we prevent the mixing of oxygenated blood from the deoxygenated blood? See, in case if they get mixed with the deoxygenated blood, then the blood pressure will not be under control, and even we will not have constant body temperature, and it will affect our immunity also, and then it will affect our immunity also. Okay, that is one reason. It prevents the blood from mixing with each other, right? And then this, it will fulfill the constant use of energy to maintain their body temperature. So maintaining constant body temperature is very important. See, you look at reptiles, amphibian and all that, they have just 3 chamber. Reptiles and amphibian, they have 3 chamber. Over there, the oxygenated blood will get mixed with the deoxygenated one. They get mixed with the deoxygenated one. That's why, you know, those reptiles and amphibian and all, they are okay with it, because they adjust their body temperature according to their surrounding. Okay, so it is not constant for them, whereas for us it is constant. For us, birds, mammals and all, it is constant. Why? Because there is fourth chamber, and it will not let the oxygenated blood to get mixed with the deoxygenated one. Anurag says it is 37 degrees, yes. Okay, that is one thing, and also the efficiency of supplying oxygen will also be high. Okay, clear with that? Fine. So next question. What are the modes of excretion in plants? So I have already covered, right, that plant also excrete the waste material. For example, they don't require carbon dioxide, right? After respiration, the carbon dioxide is released, and that carbon dioxide is thrown out into the environment. And then during photosynthesis, after photosynthesis, the plant release oxygen into the environment. Plant, when they don't require oxygen, it is released out into the environment, and carbon dioxide also, it is released out into the environment after respiration, right? And also, you know, during transpiration also, the excess water will get evaporated through stomata, right? And then the organic waste of the plants are also it is stored in some of the dead permanent tissues, okay, like wood, leaf and all that. And then the older leaf, you would have observed, right, the older leaf also, they will have waste material, and then after some time they will dry off and they will fall, right? Like that the waste material is released out from the plant body. And also some of the waste material are also stored in the vacuole as well. They are also stored in the vacuole as well, and some of the waste material you will see, right, you will notice, no, older trees, they will release those resin, gums and all that. Those resin and gums will also have certain waste material which is not needed for the plant, which is not needed for the tree. That's how plant also excrete the waste material. Okay, clear with that? Fine. So let's move on to next question. Already this question was there, the first question, right? So that is, you will have discussed the mode of nutrition in amoeba. Okay. So like I told you, first the amoeba, when the food particle is near, what will it do? It will engulf that food particle with the help of pseudopodia, right? That is called as ingestion, and then food vacuole is formed wherein the complete digestion of food takes place, and after the digestion of food, then what happens? Absorption and assimilation takes place, wherein the food, the digested food is distributed, wherein the digested food is distributed within the cell, right? And then that digested, after the complete, assimilation of that food, then what happens? There will be certain waste material, and that waste material will be thrown out of the cell through egestion. Okay. So that's what is being explained over here. The first step is formation of pseudopodia, which will engulf the food, and then there is egestion taking place, digestion and also assimilation. You should mention digestion. After that, absorption and assimilation takes place, and then the exocytosis. Okay, clear with that? Fine. So next question, what is the role of acid in the stomach? So you all know, right, that the stomach, it has got gastric glands. Those gastric gland, it will secrete certain digestive fluid which will have hydrochloric acid, mucus, and also what? Protein digestive enzyme, that is pepsin, right? So now the question is, what is that HCl doing in the stomach? What is the function of HCl? You all know that the HCl, it will kill the microorganism that is present in the food. Okay, and then it makes the environment in the stomach acidic. It makes it acidic, and that acidic environment is required for the activation of the enzyme pepsin. If the environment is not acidic in the stomach, then the pepsin will remain inactive. If it remains inactive, then there is no digestion of protein. Okay, that's why first, in order to activate the pepsin, the HCl is also secreted. So the functioning of HCl is, one is it activates the enzyme pepsin, and then it kills the bacterial microorganism, that is harmful microorganism that is present in the food, and then it also prevents the fermentation of the food. Okay, clear with that? Fine. So almost we are at the end of this biology session. This is the last question, I guess, right? So yeah, what is peristaltic movement? And then question B says, stomata remains closed in desert plants during daytime, and how do they do photosynthesis is the question, right? So now what is peristaltic movement? See, I have told you now that you eat, after we eat food, first in buccal cavity the food gets mixed, it will get mixed up with the saliva, right? After that what happens, in saliva there is salivary amylase which will break down the starch into maltose, right? And after that we swallow that food, and then that food reaches the stomach, right? So that time how is that food reaching the stomach? See, such type of movement is observed in the food pipe, not just food pipe, in the entire digestive system. Such type of movement is noticed, that is called as peristaltic movement, whereas there is contraction and relaxation is there. Contraction and relaxation is happening, because of which the food is pushed in, and that contraction and relaxation, that movement, that is called as peristaltic movement. See here, the relaxation of the gut muscle to move the partially digested food where? Downward through the alimentary canal, that is called as peristaltic movement. And then second question was, why the stomata remain closed during daytime, right, in the desert plants. Then how do photosynthesis take place is the question. So the thing is, in desert plants, what happens is that during night time they keep the stomata open during night time. So that time carbon dioxide will be absorbed. They will enter into the leaves will the stomata will be open and the carbon dioxide will enter inside, and that carbon dioxide will be stored in the leaf during night time, and during daytime the stomata will get closed, and then during daytime there is sunlight. Utilizing that sunlight, the photosynthesis takes place in the desert plant. See, like you can see over here, during night time the stomata is kept open and carbon dioxide is absorbed, and during daytime the stomata is closed and photosynthesis is happening. Okay, clear? So in desert plants, see, it is kept open. The stomata is open at night, carbon dioxide is absorbed during night time, and then the photosynthesis takes place during daytime. Okay, clear with that, students? So this is it from your biology section for today, right? So now the chemistry sir will continue the session, right? Okay, so now you have any doubts, students? You have any doubts? Okay, you want me to explain about hemoglobin? See, hemoglobin, it is, students, this is Navya. I am Navya and Neha ma'am is for physics. Okay, see again in comment section I can see some Neha ma'am, somebody telling Neha ma'am. See, they only have one doubt, the hemoglobin thing. I'll just tell you, stay here only till then. So yeah, hemoglobin, it is a iron-rich protein which is present in red blood cell. It is because of the presence of that iron-rich protein only, and that hemoglobin, it is built in such a way that it has got strong affirmation towards the oxygen. That's why it carries the oxygen, and when it binds to oxygen, the hemoglobin, it turns red. Okay, hemoglobin, it is present in red blood cell. Okay, students, clear? Now your chemistry sir is here, and the chemistry, he will explain.
Yeah, ma'am. Thank you, ma'am. Hey, everyone. How are you all? I hope everyone is safe and doing good. Okay, students. Okay, let's start. Let's start chemistry. Okay. Okay, ma'am. Bye. Yes. Biology is clear. I hope everyone biology is clear. Let's start with chemistry, students. First question. First question is we need to identify the type of reaction in by using this image. Okay, students, what happened here? From A compound, one pair of electron is removed and it becomes positive charge, and this same at a time B compound, the releasing pair of electron is there now. It accepts that electron and it becomes negative charge. We have four options, students. First option is redox reaction. Option B, they mention combination reaction. Option C, they give decomposition reaction and displacement reaction. What is the answer? Come on, students. Let me know on chart section first. First, it's an easy reaction, students. Come on, let me know. Hi, Varad. Let me know, students, fast. Let me know. What is the answer? Come on, students, answer it fast. What is the answer for this question? Let me know. Fast. Student, your Suhasar is waiting. The answer is redox reaction, right? For redox reaction, we have three types of definition. In first one, it takes place either oxygen is added, or from the one molecule oxygen is added, and similarly in other molecule oxygen is removed, and also from electron pair also we define if one compound at the same time one compound is lost its valence electron, it is called as an oxidation, and in same reaction one compound receives the pair of electron and becomes negative charge, it is called reduction reaction, right, student? The redox reaction, no, not in displacement Gagan, it's a redox reaction. The answer is redox reaction. Why? Because from electron also we can define the redox reaction. In this what happened, reduction and oxidation takes place at the same time. It's clear? I hope it's clear, students. It's clear? Gagan is clear? It's a redox reaction. Okay, let me move on to the second question. The second one is a white precipitate formed by the reaction of barium chloride. BaCl2 is reacted with sodium sulfate. Is nothing but Na2SO4, right? Na2SO4, its forms, they only give, if white PPT is formed, they give 4 options, right students? In option A, they give BaSO3. In option B, they give BaSO4. In option C, BaO and BaS. What is the answer? We already know these two are wrong answers. I ruled out this answer, this option, and the answer may be A or B, right? B. I use B. And what's about other students? What's about others? Yes, I use is right answer, right? It is called double displacement reaction. What happened? Ba is replaced by sodium and sodium is replaced by Ba. What happened? It forms a white precipitate with the formula of BaSO4, barium sulfate and NaCl. To balance the reaction, we need to mention 2 in RHS side. Right, students? Right, students? B is a correct answer. I hope it's clear. It is called double displacement reaction. From the unit, I already explained you guys. This is a very important topic, right? In this topic, we can expect, not an expect, definitely they ask one MCQ from double displacement reaction part. Especially this example. Most of the question paper, the example is repeated. The question format is different, students. Great way of teaching. Thank you so much, Ayush. The question, what? The way he ask na, it's different. For example, sometimes they give the reaction and they give 4 options. Mention the type of reaction or what is the type of reaction or what is the type of reaction in given example. That thing they ask. Right, students? Thank you, Ayush. Let's move to the next question. Which of the following is not a mineral acid, students? Please read the question twice or thrice in examination also. We already know when exam we are in array situation. Instead of not, we read is A. So many times, so many students do this mistakes. Please, sir, question par zoom karo. It can't able to zoom Gaurav, sorry. No, sorry, Gagan, it can't able to zoom. I explain once. Which of the following is not a mineral acid? Is not a mineral acid, student. Not a mineral acid. Okay. They give 4 options. In option A, they give nitric acid. Is nothing but HNO3. Right. Second one, sulfuric acid. Okay. H2SO4. In option C, they give HCl. Okay. In option D, they give citric acid. What is the answer? Mineral acid is nothing but they are dissolved in water, but they are not dissolved in organic solvent. Right, students? Sir, please read what? Read done. No, students, sorry, I can't able to zoom. Gaurav, sorry. I explain. I read the question and I write it once also. Okay, now? Okay, students, what is the answer? We need to identify non-mineral acid, students. Citric. Yes, it's a right answer. Ayush, what's about other? What's about Gaurav? Yes, Gagan is a right answer. Gaurav. Yes, yeah. Citric acid. Why, students? Yes, it's a right answer. Citric acid. These three acid, hydrogen compound is present, and it is these 3 all dissolved in water. What happened? Intermolecular hydrogen bond is formed these 3 acid, students, but except citric acid. Citric acid is an non-mineral acid. It is dissolved in organic solvent, students, not in water. It's clear? Okay? That's why if we already know citric acid is present in lemon, right? If we mix the lemon water, what happened? The color of the water is changed. When we mix the HNO3 or it gives different solution. Okay? Okay, students. I hope it's clear. Let's move on to the next question. The next question is substance X. One substance is formed a reaction of carboxylic acid and alcohol. If carboxylic acid COOH group and OH group is reacted, it gives X compound. Okay, this X compound we used it. It is used in making ice cream. Wow, ice cream, right? We already know about it. Okay, it gives ice cream, cold drinks also, and also perfumes and flavoring agent. In many criteria we use this X material or X substance formed by the reaction of carboxylic acid and alcoholic group. We need to identify X, right? Okay, they give 4 options. Students, please concentrate on question, please do this 1L for your Suhas sir. Please concentrate. This 20 days is a very important days, guys. Please concentrate. Please stay with us. Please, everyone, I ask you. I apologize everything. Sorry. Please do this. Okay, student. Gaurav, stop it, please. For your Suhas sir, please do this help. Gaurav and also Gagan, please answer the question. Please concentrate on the question, students. Please do this 1L for your Suhas sir. Please, guys. Okay, they give 4 options, students. First one is an aldehyde. Option B, they give alkyne. Option C, ester. They give ester. Option D, ketone. What is the answer? Gagan, aldehyde. Gagan, please read the question. Please read it and then answer. Okay, what about Gaurav? What is the answer? Ayush, what is the answer? C, ester. Ayush answer is ester. S. It's a right answer, Ayush. The reaction between carboxylic acid and alcohol in the presence of any acid, any medium is called esterification reaction. What happened here? Ester group is formed. Ester is nothing but C double bond O group, right? This group is formed. This reaction we called as an ester. It's clear? Students, here one important point is there. Ester is clear. S Gagan is an right answer. S Ayush is an right answer. Gaurav. Okay, students, understood? Gaurav, please answer it. Understood, it's very important. In explaining class also, Ankita ma'am told this reaction is very important. I also explained the top 10 important topic, right? Ethyl ethanate. S Gaurav is an right answer. Ethanate is nothing but ester compound. Students, this compound CO bond we called ether bond, but ether is nothing but ethoxide. Okay, ethoxide is called ether. Similarly, C double bond O compound is called ethonate. It's clear? Please concentrate on this naming. Okay, if instead of, in the place of metal, if we reacted with metal, what happened? It gives different. Okay. Yes, Ayush, thank you. I hope it's clear, students. Let's move to the next question. It's a very important. Most of the question paper, in most PYQ question, this question is there, right? This question is from metals and non-metals unit. Where's the pen? Yes, M and NM. Okay, let's read the question. The question is, take 3 boiling tubes. Here 3 boiling tubes is there. Pour some water in test tube A. In test tube A, there is only water with air is there, students. In test tube B, pour boiled distilled water. This water is already boiled water. And in another test tube B and other also oil is there, students. And next in test tube C, put some anhydrous CaCl2, calcium chloride. Okay. The question is, leave all the 3 test tubes for 1 day, then observe. We need to leave all the test tube. Okay. We have 4 options. The question is clear, students. The question is, in which test tube nail is rested? We need to identify in which test tube the nail get rested. Okay. We have 3 test tube. It's clear, students. Question. In first test tube, nail with water and air. In second test tube, water plus oil is there. In test tube C, we only anhydrous CaCl2. They give 4 options. In option A, they mentioned tube B and C. Option B, tube B. Option C, tube A. Option D, tube A and C. What is the answer, students? Aish, what is the answer? Gaurav? And Gagan, what is the answer? Let me know on chart section, students, fast. Your Suhas sir is waiting, students, fast, fast. A. A is an tube, B and C. We need to identify only in which test tube the nail is rested. In tube B, oil is there, Gaurav. Please read the question. Oil is also there in tube B. In tube C, only another CaCl2 there. What is the answer? Okay, I think tube A. Right, Gaurav? Tube A. Yes, it's a right answer. The answer is option C. Tube A. Okay, it's clear. The answer is tube A. Why? Here we already know for rusting. Yes, C. Yes, it's a right answer, Gaurav. Okay, we already know for rusting both moisture and air both are important. We already discussed with exercise in acid, base and salt also, metals and non-metals unit also. Yes, Gagan, it's a right answer. See, what happened here? The oil breaks the connection between water and air. That's why it's not rusted. What is the role of anhydride CaCl2? It is only what? It is only absorb the moisture around the surroundings. In surroundings some moisture is there now. This CaCl2, anhydride CaCl2 is nothing but here water molecule is low. It is no water molecule is concerned. It is there. That's why. Understood? It's clear? I hope it's clear, students. If it's clear, type CC in chat section fast. Next question. The sixth question, students. We already know from chemistry, according to CBSE board, totally 8 MCQ, 1 2 marks question, 1 3 marks question for 2 and 3, not in 2, only 3. We have option also there for another one 5 marks question. This 5 marks question completely based on the unit CB, carbon and its compounds. Okay, C and C. This 3 marks from metals and non-metals unit, and also one case-based study question. This case-based study question according to CBSE data sheet. Not a data sheet. According to CBSE mass distribution, this case-based studies, it's definitely they ask from the unit metals and non-metals or acid, base and salt. From these two units, we can expect these case-based study question. It's clear? B. Okay. The question is the ratio of HCl and HNO3 in aqua solution. Aqua is almost all students are answered. Ayush, Gagan, everyone answered B. As students, it's a right answer. 3:1 ratio. Where this aqua solution is now we use it. We already know the low reactivity metals is nothing but the gold metal, right? Gold and platinum and also sometimes silver metal. These three metals we called as a noble metals. Why? These three metals, they never reacted with oxygen, right, students? Expect this silver. Sometimes it's reacted with sulfide compound to form Ag2S, silver sulfide, right, students? But these two metals never reacted with oxygen or anything. Sir, you are the best. Thank you so much, Gaurav. Thank you so much for your appreciation. Right, students? These are what? These are noble metals. That's why these metals we can easily obtain as an elemental form. They never reacted. For reacting, these metals also reacting, we can easily dissolve this metal using aqua regia. The aqua regia solution is an 3:1 ratio solutions of the HCl and HNO3. If we dip the gold in this reaction, what happened? The upper surface of the gold, copper is also low reactive, Gagan, but it is reacted with oxygen compound to form copper oxide. We already familiar this, right? CuO compound. But except these two metals is there now, gold and platinum. These two metals never reacted. Okay? Okay, students? I hope it's clear. If we dip the gold in this aqua regia, its upper surface is dissolved in this solution, either in concentrated HNO3 part or concentrated H2SO4 part. We already is familiar. Let's move to the assertion and reason based question, students. The question is assertion. Copper vessels get covered with green coating in rainy season. In rainy season, what happened? Copper metal, Cu metal is covered with one green color vessels. Okay, green color vessel. Because the reason is, it is because of the formation of copper carbonate. Okay, we have 4 options. For every options, these are same, right? For case based, not in case based study, assertion and reason, the options are same. In option A they give both A and R are true and R is the correct explanation of A. Both A and R are true but R is not correct explanation. Option C they give A is true but R is false. Option D they give A is false but R is true. What is the answer? A. Gaurav answer is A. Gagan answer is A. Sorry, B. Answer, what is the answer? Ayush, what is the answer? As Gaurav, your answer is right. The answer is option A only. Yes, absolutely. Copper vessels covered with green coating. What happened? The atmospheric oxygen is removed and that's why the answer is option A. It's clear, students. This is an answer for the question. Ayush, C, not Ayush is an answer is option A. Okay. Let's move on to the 2 marks question, students. Here MCQ is over already. Any doubts about MCQ questions? If you want any explanation about MCQ question, students, and I also tell something for you. In tomorrow, I take the pair of linear variables in 2 equations. Okay, please attend the class, mathematics class, and also day after tomorrow is nothing but Tuesday, and Tuesday we start ABS. ABS means acid, base and salts unit with one shot and with PYQ questions. Okay, please attend it both classes. Okay. Okay, students, I explain the question. Okay, what happened? The atmospheric oxygen attacked the copper and it forms and covered with one green coating, right? Okay, and also reason what they give, it is because of the formation of copper carbonate. Absolutely. What happened? It's attack in atmospheric, carbon dioxide is also there, oxygen is also there. If the copper first time is attacked with oxygen, it to form copper oxide with, then it attacked with CO2 compound and to form CuCO3, copper carbonate compound, CuCO3 compound. It's clear? Gagana, I hope it's clear. Let's move to the next 2 marks question, students. The next question is, a gas is evolved when ethanol, what is the molecular formula of ethanol? C2H5OH, right? With sodium, what happened? Any metal is reacted with ethanol, it gives one compound and also one gas is evolved. We need to identify. Akshit, sir, today my father friend's son Vishwe's birthday, please wish him and bless him. Yes, absolutely. Happy birthday, Vishwas. Happy birthday. Hydrogen. Hi, Mr. Superbeep Gaming. Hydrogen acids and right answer. As we need to write the, and also the question is name the gas evolved, and also we need to write the balanced chemical equation for the reaction involved, students. We are already familiar this reaction. It is most important reaction. So many times they ask, right? What happened? We already know sodium is highly reactive than hydrogen. The sodium reacted with ethanol compound. This hydrogen gas is liberated, right? 2 molecules. We need to balance the chemical reaction also. It's very, very important to form sodium ethonate compound. Sodium ethoxide, not an ethonate, right? Sodium ethoxide. Sodium ethoxide compound with a liberation of hydrogen gas. How to confirm the liberation of hydrogen gas, students? 2 compound. It's clear. How to confirm? CO2, not an CO2. It's an H2 gas. As Mr. Sup, it's an right answer. As it's an, the reaction is right, students. Right, super. Kudos for you, Gaurav, and also Mr. Super B Gaming. Kudos for you also. Okay. The answer is sodium ethoxide is formed. Ethoxide is nothing but here CO bond is formed. Ether. It's formed an ether compound, not an ester compound. Okay. It's clear. I hope it's clear. How to confirm this gas? By using pop-up sound. Right? If we take a matchstick near the hydrogen gas, it sounds a pop-up sound. I hope it's clear. Hi, what Pilupari, hi, pop sound, as it's an right answer. Okay, student, it's a 2 marks question. How to write the answer for this question? First, we need to mention the gas in the particular reaction. Hydrogen gas is liberated, right? Hydrogen gas is liberated. Next, we need to mention the reaction next. This is a reaction, and also students, please confirm us. The reaction must and should be balanced. If it's not balanced, definitely you lose the marks. You lose the marks, and you need to naming the compound. You need to name the reaction or name the compound below the reaction. For example, here ethanol, and here sodium, then sodium ethoxide, and please concentrate on naming. We have confused with CO bond and C double bond, ester compound, ether compound. For ester we name sodium ethonate compound. In the place of ether, in ether we name as an ethoxide compound. It's clear, students? Bacho, it's clear? It's clear? Students, please maintain a discipline or neatness in the question answer sheet. If you maintain neatness, you definitely score 80 out of 80 in your board exam. Stay with us. Definitely you guys will score the marks. Let's move on to the next question, guys. Yes, the next question is, shall we move? Yes, a group of students. For example, there is a one bunch of students or there, group of students or there. Look at different metals and metal sulfate solution. Given is it tabular form? What tabular form? We have here metal and metal sulfate solution, and also they mention their color also, students. The question is clearly visible. I hope it's visible, students. In this, form the data, answer the following. We need to answer these 3 questions. It's a 3 marks question. For every questions, they give 1 marks. This is a marks distribution. Metal. First metal is an chromium, cobalt metal, copper metal, magnesium metal. They give. For chromium, the respective salt, the corresponding salt is chromium sulfate. For cobalt, the corresponding salt is cobalt sulfate. Copper, copper sulfate. Magnesium, it's a magnesium sulfate, students. These are the color. These are the respective color or the corresponding color. For chromium sulfate, it has a green color. Cobalt sulfate, pink color. Copper sulfate, we already familiar this color, blue color. And magnesium sulfate is a colorless. It's a clear solution. Right, students? The first question is, the first question in this exercise. Let's check it, students. Gagan, please, Gaurav, stop it, guys. Please concentrate on question. Why, guys? Please, Gagan, for your Suhasar, do one help. Please concentrate. Please answer it. And next, whatever can you do, guys, after session, please concentrate. Please answer it, guys. Please. This is request from your Suhasar. You need request from your Suhasar, na? You want that? Please answer it, students, please, for your Suhasar. First question is, which metal reacts with all other sulfate solution? It's nothing but, we already know, the metal is highly reactive. It reacts with all solution, right? It reacts with all the other solution. We need to identify the high reactive metal. We already know about the reactivity series, everything. We divided the reactivity series into 3 parts. The decreasing order of the metal reactivity we call what? Reactivity series. For example, we divided into first highly reactive, second moderately reactive metals, last one we call least or low reactive metal, and also some metals we call as a noble metal. Right? Right, students. First question, which metal reacts with all other sulfate? Please let me know an answer. Displacement reaction will happen as Gagan. Thank you so much, guys. Please understand. Gaurav, answer it, Gaurav, and also Gagan, please. Okay, fine. We need to understand, we need to clear here displacement reaction is occur. It's a very good point, students. It's a best, and also it is a correct reason for the question. But we need to identify the metal. Here let's check chromium, where chromium is present in the reactivity series. In the video top 10 most important topics, students, I give one simple trick to remember reactivity series. Please check it once, okay? After this session, please check it one. Not visible. Okay, students, here 4 metals is there. First metal is in chromium metal, Cr metal, cobalt metal, Co metal, copper metal, Cu metal, and magnesium. Which metal is highly reactive in these 4 metals? Which metal, Gagan? Which metal, Gaurav? Which metal, Pari? Yes, the answer is, we already know, compared to these metals, magnesium is an highly reactive metal. magnesium, aluminium. First metal is in potassium, sodium, magnesium, aluminium. This is a series, right? These metals are called, we call as an highly reactive metals. This magnesium is reacted with oxygen easily to form magnesium oxide in hot condition. We already familiar. Okay, answer is magnesium. I hope it's clear. Second question. MgS, Paris and right answer. Second question, we need to identify which metal did not react with any other metal sulfate solution. It's nothing but the metal is must and should be low reactive. Low reactive in this compound, which is the low reactive metal. We are already familiar with this copper metal, right students? So many reaction. What happens? Simply the high reactive metals is come and pushed out to outside, push the metal into outside, like an in WWE the Royal Rumble is happens now. Some Big Shaw is coming, some other what opposite players is coming now. They simply pushed out. Similarly, the highly reactive metal pushed out the copper in the reaction or copper in the solution to form a new bond with sulfate solution. What happened? That's why the answer is copper. Copper Cu compound. For example, let's take magnesium compound. Copper sulfate CuH44. Both are +2. This magnesium is pushed out copper from copper sulfate, then it forms magnesium sulfate solution, and also copper get eliminated. It's also one method to extract the copper from its ore or from its metal solution, right? It is also one method. Okay. Next, last question, students. We need to arrange the metals in decreasing order, not a increasing order. Okay. Think decreasing order, not the increasing order. Decreasing order is nothing but we need to mention with decreasing symbol. Let's change the pen, color of the pen. Let me give white pen, okay? Yeah, we already know magnesium is highly reactive. First magnesium is come. Last copper is come. It's clear. We need to differentiate between chromium and cobalt. Compared to cobalt, chromium valency is little bit high. It's as +2, cobalt +3. We already know. That's why chromium is, compared to cobalt, chromium is highly reactive. This is an order, decreasing order. Yes? Gagan is clear? Everyone, I hope it's clear. Okay, let's start. Let's move to the next question, guys. The next question is an organic compound A. We have an organic compound A. It is a constituent of many medicine. It is used in many medicine and it is used as an antifreeze. It is used as an antifreeze compound. Okay, students. And also they give molecular formula of A. What is the molecular formula of A? It's C2H6O. C2, carbon 2 molecule is there. Either the compound may be ethanol or ethanoic acid. Ethanol or ethanoic acid. Then this question is coming from the unit. Which unit, students? Carbon and its compound. It's a 5 marks question. Clear? Yes, thank you so much, Gagan. Then this one is upon reaction with alkaline, alcoholic KMnO4. It's reacted with alcoholic KMnO4 to yield compound A is oxidized. This compound A get undergo oxidized, students, to give a compound B with molecular formula C2H4O2. C2H4O2. We need to identify the compound A and B. We need to identify these two compounds and also write the chemical equation for the reaction which leads to formation of B. It's a very easy reaction. Right, oxidation reaction. If alcohol, they give alcoholic KMnO4, it's an oxidizing agent. This reaction helps to what? It helps to oxidize the compound to give respective products. We are already familiar this reaction. A. In A, what is the molecular formula? In A, C2H6O is there. C2H6O. Let's write ethanol compound. C2H5OH. A is nothing but ethanol. It's an oxidation process, right? alcoholic KMnO4 acts as an oxidizing agent. What happened? On another O molecule is added, the two H molecule get eliminated. In H6 compound, it becomes C2H4OH, CH3COOH compound. It's an ethyl ethanoic acid or acetic acid. We are already familiar, students. These two substances there na, ethanol and ethanoic acid, these two are very very important, especially that saponification reaction, esterification reaction, and also this oxidation reaction, dehydration reaction. We already know, familiar with dehydrating agent also, and also soap and detergent, formation of scum, and also the question how they ask in soap and detergents, especially they ask like, we cannot, the soap is not, it is not suitable for use for hot water. Why? Because it forms a scum. It reacts, students, it reacts. What it reacts? Soap is react with what soap is used in hot water. It react with, we already know hot water contains magnesium and other minerals, right? It's reacted with and it forms a scum. These are very important. Sir, आप South Indian हो, please बताओ. ऐसा, I am a South Indian. I am in Karnataka. I am living in Karnataka, especially in Bangalore. Okay, it's clear? Next, move to the next question. It's a very interesting and very easiest question. The question look like and so long, but the answer is small only. Let's check. Okay, let's check. Come on, students. Let's check it, students, fast. The question is, M is an element which may be out of CuFeAlNa. M is an element. Any element, they only give 4, 1, right? Aluminium and Na. It shows the following property. First property, one of its 4 is M2O3. Its 4 is an M2O3. M2O3 is not affected by water. It is not affected. M2O3 is not affected. This 4 is not affected. Clear? Option C, it corrode easily. It can easily corrode, undergo rusting easily. Then last one, it forms 2 chlorides. It forms a chloride in the form of MCl2 and also MCl3. We need to identify M, right? Let's check it. Copper. What is the valency of copper? 2 plus. If copper is reacted with oxygen, it gives UO compound, not an M2O3. That's why copper is eliminated. Sodium. What is the valency of sodium? Plus 1. If it's reacted with oxygen, it gives an NaO2 compound. It's not obtained in the M2O3 form. Al. Bauxite, yes, Al2O3. It's obtained as bauxite ore. Yes, it's obtained. And also, let's move on to the iron compound. As Fe2O3. Iron is also obtained. Okay, then it's nothing but these two compounds are eliminated. These two elements, copper element and sodium element already eliminated. The answer may be iron or aluminium. But finally we need to identify only one compound. Already Pari is identified as it's a right answer, Pari. Fe, yes, it's a right answer. Second, M2O3 is not affected by water. Both are not affected by water. It affected by steam only. We are already familiar that reaction in metals and non-metals. In most of the reaction we already discussed. Metal oxide, the reaction of metal oxide with water, we already discussed that it's not affected by water. Both are, it corrodes easily, right? Yes, absolutely. Aluminium is also corrode easily. Iron is also corrodes easily. Both are corrodes easily. That's why the alloy compound is alloys are used. Why? To increase the property of metals. We are already familiar this alloy, aluminium alloy also, iron alloy also, right? That's why we steel are used in many process. But last property gives the answer. It forms 2 chlorides, both MCl2 and also MCl3. But aluminium can't form 2 chlorides, but iron, it can able to form 2 chloride. How, sir? How can it possible, sir? How can it possible, Suhas, sir? As it's possible. Why? Depends upon the counter ion. It changes its valency. Sometime we already D, iron is coming for D series. Sometime what happen now? we already know we have two valencies, one halfly filled, it is also stable compound, and fully filled. Depends upon their, yes, two valencies, S is an right answer, PU is an right answer, it forms two valencies, okay. If you go through higher studies, in PU, I already teaching PU also students, +1 and +2, it is there, D block components is there. D block compounds, you study this concept. That's why the answer is iron. This is an competency based question, student. It's nothing but, it's a 3 marks question. Could you believe guys, if this is a 3 marks question, if you write iron, you got 3 marks. Sir, have a doubt? S please, S, result S. Sir, what is iron 2, 3 and zinc, copper, what is the 2, 3 numbering? I am confused. Okay, you are confused. 1 second, I explained it. 1 second for Krish. Okay, what happened, Krish? We need to identify the metal. Copper, we already know. The valency of copper is 2 plus. That's why if it reacts with oxygen, it forms CuO. But the ore is in the form of M2O3. That's why sodium and copper get eliminated from first property only. Second property both are follows, and third property is also follows. Finally, iron. Iron, it has a 2 valency, 2, 3. It has a 2 valency. Sir, easy but mushkil. Yes, ferric and ferrous is the right answer. For 2 plus we call ferric and ferrous. Depends upon the counter ion. Aqueous solution and liquid. Students, aqueous solution is nothing but it's a solution. Solution and liquid is a different. Liquid is only one component. Solution is a mixing of two components. It's clear? Result is clear? Aqueous solution is nothing but in aqueous solution we have two components. Liquid, only one component. For example, ethanol is a liquid, right? But sodium hydroxide is an aqueous solution. What happened? NaOH is dissolved in water, students. That's why we called aqueous. But liquid is nothing but only one component. It is in the form of liquid. Then why H2O is liquid? It's clear? Okay, once again I repeated result. Okay, water is a liquid. Two components is nothing but aqueous is nothing but one compound is dissolved in another solvent or solution. Okay, one solvent is dissolved in another. Sir, you don't know Hindi, only for knowledge. Students, I understood Hindi, but I can't able to communicate with Hindi. Why? Because I am a South India guy. Okay, that's why I communicate with you English. It's clear? I am familiar with Kannada, students. I have a 6 months plus experience in Kannada. It's clear. The answer is clear. Anna, S, insoluble. Anna, S, Tamma. Yes, okay, it's okay. It's clear. I hope the question is clear. Last question, students. After this question, Neha ma'am, she is waiting. She come and explain the physics concept. This is an CB question, case-based studies question. Okay, sir. Oh, thank you so much, Krishu. Thank you. The question is simple. The this case-based study question from the unit ABS. ABS is not about the apps. It's about acid, base and salt. Okay. I hope. Let's move to the question. The question is very simple. Salt of a strong acid and strong base is in neutral. If a strong acid, for example, for example, as HCl and NaOH, if strong acid is reacted with strong base, we get neutral pH value. NaCl, it has a neutral salt, right? It's clear. NaCl is a common salt, is formed by a combination of HCl and sodium hydroxide. I already write the reaction. This is the salt that is used in food. Common salt, NaCl is used in food. Chapter 2. Yes, it's a right answer. Pari, in Tuesday, I take ABS one shot within 1 hour. I plan to cover the overall unit in 1 hour or 1 hour 30 minutes, and we discuss some more PYQs. Please attend it. Phosphoric acid. Oh, you already tell the answer. Okay, let's go. Some salt is called a rock salt. We already familiar. This rock salt, bit of rock salt was formed when seas of bygone gas dried up. Rock salts are obtained in the seas, right? Yes. The common salt thus obtained is an important raw material for various materials of daily use, absolutely, such as sodium hydroxide, baking soda, washing soda and bleaching powder. We use it in various. The question is, case-based study question is nothing but the question is only related, but the answer is never in this case. It is not there in this case. So many students are thinking, oh, case-based study, we do not read the question. The answer is only in what paragraph, but both are different students. Okay, it's clear. The question is, if given acids are phosphoric acid, carbonic acid, hydrochloric acid and sulfuric acid, then which acid does not form acidic salt? Which acid is formed acidic salt, students? If the weak base is reacted with a strong acid, it form acidic salt. Here carbonic acid, hydrochloric acid, HCl and H2SO4, it forms a strong acid, acidic salts only. Why? Because both are strong acid. H2SO4 is a king of acid. First one, phosphoric acid, does not form salt. It's a right answer. Kishu, kudos for you guys. Okay, what's about others? The answer is first one, the answer is phosphoric acid. It forms a basic salt, student. Phosphoric acid forms a basic salt but not an acidic salt. It's clear. Second one, what is the formula of baking soda? We already familiar this baking soda in many reaction NaHCO3, right? The R question is also there. The R question is, name the substance which on treatment with chlorine to obtain bleaching powder. Which substance? HCO2 has CaOH2, students, not an HCO2. CaOH2, calcium hydroxide, we use it in the treatment of chlorine to obtain this one. Let's know the answer. One second, students. This is an answer. Okay, it's clear. The answer is this one. Calcium hydroxide, we use this calcium hydroxide is react with chlorine to form bleaching powder. It's clear. I hope it's clear. If you have any doubt, let me know on chat section. First, chemistry is clear. This about today's sample paper. If you have any doubt, let me know on chat section. Neha ma'am is coming. Come ma'am. Yes, thank you, student. If you have 1 second, ma'am. Yeah. Any doubts, students? First, I hope no doubts. Okay, if you have any doubt, let me know on comment section. Neha ma'am is starting. Okay. Okay, the result doubt is there. Okay, the doubt is, sir, if we add common salt in water, it would be dissolved and nothing would happen. As it's an right answer is not an, yes. If a physical or chemical change, it is an physical change, students. It's not a chemical change. There is no new product is formed. It's dissolved. Okay. Okay, students. I hope it's clear.
Okay, so hello students, can you hear me? Yeah, can you hear me? Just tell me quickly, can you hear me? Yeah, shall we start with the physics questions? Yes, shall we start? Okay, I hope the audio is proper. So done, we are going to start with the physics questions now. So you are done with bio and chemistry questions, right? Yes, we are starting with physics question. Let's enter, let's see what are the physics questions that we have today. Okay, in the morning session we have, yes, so let's start today's session of physics. Okay, hi to everyone. So in the morning session we have been discussing about the sample paper 1, right? So yeah, now we are going to solve the sample paper 2, right? Yes. Yeah, light chapter will be covered by tomorrow, okay? Tomorrow, Monday, I will be covering the refraction part. No worries about that, okay? Today, we are solving the sample papers, right? So, the refraction chapter, that is of your light, will be covered tomorrow. Don't worry. Yes? So, let me display the first question without wasting much time, okay? Let's solve the questions. So first question is, what does the tangent at any point on the magnetic field line indicate? Yeah, so what's the answer? Tangent at any point on the magnetic field lines, what they indicate? Okay, it will, is it direction of magnetic field, direction of force, direction of current, or direction of induced current? What's the answer? Yeah, what's the difference between, Krishu, what's the difference between dispersion and scattering? Dispersion means it will split into 7 colors, but scattering means the light ray just scatters everywhere. It will not split into 7 colors particularly. Sorry. Yeah. A and C. So what's the right answer? Direction of, see here, magnetic field line. If you draw any tangent on the magnetic field line, you will get nothing but the direction of magnetic field. Right? It's option A. Yes, Krishu, it's right. No worries. Those who didn't answer, you can try. Yes? Yeah. So the guys who, so you people are watching the video, just type down the answer, okay? Yeah, I want students interaction, I want your answer, that's all. Okay? Is this clear? So let me give you the answer here. So you can see on your screen, it's option A, right answer. Why? The tangent indicates the magnetic field. Yes, so we use it as alternative to compass. Why do we use compass? Yes, okay. Then, yeah. Keep answering to the questions. That will be great. Okay? Yeah. So what I said, when we draw a tangent to the magnetic field, we get the direction. So we use compass to get the direction, right? Why do we use compass? It will be pointing towards North Pole and other directions, South Pole, etc. to show us the direction of magnetic field. Okay? In the absence of compass, we can use this method to find the magnetic field. Is that clear? Shall we move to the next question? Yes, shall we move? Okay. Yeah, let's move to the next question that is on your screen. And what's the question? If the resistance of certain copper wire is 1 ohm, then the resistance of similar dichrome wire will be dash. What's the answer? AC and DC. AC is alternating current, means the graph of current will be like this. Okay, it's AC, and DC will be like this. So it's called as alternating current, and this is direct current, only one direction. Yeah, no worries. If you want to know, you can know that. Okay, so tell me the answer here. What's the right answer here? What's the right answer? I'm waiting for the answer. Is it A, B, C or D? What's the right answer? Yes? So I'm waiting for the answers. Yes, students? Yeah? The students who are watching this video can tell the answer. What's the right answer? So let me tell the answer. So the right answer for this question is A or C. It is B, okay? This is just a fact, okay? It's not like your concept is not strong with this question. It's just the fact you have to remember. Nothing concept works here. The fact is, this nichrome wires have 60 ohm resistance more than the copper wire. This is just a fact, okay? There is nothing much concept here. You should just remember this number, that's all. Okay, so don't worry. Done? Done with this question? Yes? Yeah, 60 ohm. Right answer. Yes, okay. So yeah, shall we move to the next question? So these are MCQ questions where there is a need of explanation of concept. I'll be explaining that. Okay. These are the direct questions, so there's no need of explanation. Yeah, it's B, 60 ohm. Right, students? So shall we move further? Let's see the next question. Yeah, next question will be on your screen right now. Okay. Which of the following terms does not represent electric power in the circuit? So you should tell which formula is not related to electrical power. Yeah, what's the right answer? Yes, what's the right answer? Okay. So, yeah, I'm waiting for the answer. Yes, what's the answer? Option C. Yeah. What is the answer? V by B or yeah? Which one? Option B. Yes. So let me write down. So first of all, what's the formula to calculate power? Basic formula of power. Tell me that. The basic formula of power. P is equal to what? What's the basic formula to calculate power? C or D? Okay, let's see. So tell me quickly, what's the formula to calculate power? You have studied the chapter electricity, right? In your schools for your internals and all. V into I. Yes, power is equal to V into I. Right? And tell me, what is Ohm's law? Tell me, what's the formula of Ohm's law? Yes, quickly. Ohm's law. V square by R. Let's check. Okay, no worries. If net is slow, no worries. C is the answer. Let's check. Let's check that. Before that, tell me what's Ohm's law? V is equal to IR. Absolutely right. V is equal to IR is the Ohm's law. Now, I'll be using this Ohm's law. Okay? Yeah, current is directly proportional to potential difference. Power is equal to work done by time. Yeah. Okay. Now, I'll be using this Ohm's law to derive some more formulas here. Okay. Yeah, let's do that. So, first I'll substitute in the place of V. Okay. In the place of V, I'll substitute IR. So, one formula I'll get here is I square R. So, is there I square R? So, this is, they have asked in the question, does not represent, right? So, this is not the answer. Okay. Okay. Yeah, at constant temperature, absolutely right. So, let me now substitute in the place of I, okay? V I'll keep as it is. So, from this I can write I is equal to V by R. In the place of I'll substitute V by R, which turns out to be V square by R. Where it is? So, this is also not the right answer. And as we stated first, P is equal to V into I. This is also not the right answer. Then the right answer is option C. Constant dimension. No, it's not required. Constant temperature is needed. That's all. And what's the right answer here? It's option C, I into R square. Done with this question? Is this clear? Yeah, option C. So you guys have already answered it, right? Option C is the right answer. Very good. It's the right answer. So whenever you get this kind of question, instead of jumping directly, you can take some time and you can figure what is right or wrong. Okay. Yeah, done. Then we shall move to the next question. Yes. So yeah, what's the next question? Okay. Assertion reason type of question. So what's the assertion? The magnetic field produced by a current carrying solenoid. Okay, so they are telling about current carrying solenoid independent of its length and cross-sectional area. Okay, yeah, the magnetic field inside the solenoid is uniform. Where? Inside the solenoid. Now tell me what's the right answer here. Is the assertion true? If it is true, does they both state each other are correct explanation or not? Tell me quickly. Yes? Yes, students, what's the answer? Option A. You're telling, sorry. Yeah, R is correct explanation. Is R the correct explanation for A? Think about it. Option B. What's the right answer? The right answer is option B. Yes, both the statements are true, absolutely true. But what happens here is this uniform inside the solenoid does not state that length and cross section. You're getting my point? Yeah, it's B. Yeah, it's B. Definitely. Then what I'm telling here is both the statements are true, but this is not the correct explanation. Then what is the correct explanation? What is the correct explanation? See here. The magnetic field is independent of length and area because what is the formula to calculate magnetic field? What's the formula? Tell me quickly. B is equals to formula to calculate magnetic field. Yes. What's the formula? Yes. Can you guys tell the formula? Shall I write? B for best. Yeah. No, tell me the formula. Oh, that will be so good. If you will be top of your class, so I'll be so happy. Good. Yes, B is directly proportional to current. Yeah. So we'll write it as mu naught Ni also, right? Mu naught N into I, where N stands for number of rounds per unit length. So, yeah, yes, it's number of turns per unit length, right? But in the formula, here we are not mentioning B is directly proportional to L or it's inversely proportional to A. We are not writing that, right? So, what happens? Our magnetic field does not depend upon length and the area. Is that clear? Yes, is that clear? Magnetic field does not depend upon length and area. Right? Yes. What should I say? N is the number of turns per unit length. If you want that. So what do you want? What do you want to pronounce? Is this done? What I am telling? Magnetic field B is equals to mu naught N into I. But it is not dependent upon length and it's not depend on the area. So I shall move to the next question. Yeah? Okay. So we are done with the 1 marker question. So we have a big question here. So let me read out the question first. Done? Okay. An object is placed at the distance of 10 centimeter from a convex mirror of focal length 15 centimeter. Find the position, nature of the image. So let me first take the given terms. It's 10 centimeter, object is placed. So U is equal to 10. That too minus 10. Okay. So yeah. So but it is the standard formula, right? In magnetic effects of electric current, you will not be getting much formulas and numericals, but you must be good with the concept. To remember the concept, I gave you that. Right? Okay. Moving forward, focal length. So this is convex mirror, focal length will be positive. Now quickly calculate what is the answer. What's the answer? So what it is given, U is equals to minus 10 centimeter. Focal length is equals to 15 centimeter. It is a mirror. So I'll be using mirror formula. Okay. I'll be using mirror formula. Okay. So before solving, Can we directly say without solving this? Yes? Can we directly see this? Can you guys directly see the answer? Now I thought at a point, why it's not necessary, it's not required to solve also. Directly we can say. Directly we can say. Yes, it's behind the mirror, virtual, erect and diminished, that's all. If they ask specifically a number, we can calculate it and say. But they are asking just find the position, nature. Yeah, position, you can say that it's behind the mirror, and nature, it is diminished, it is virtual and erect. It's not required to calculate. If you want to calculate, you can calculate it. Do you want the calculation? Yes, let's calculate once, because all the time they'll not ask like this, right? Sometimes they may ask you for the image distance. So I will calculate. So mirror formula is 1 by F, 1 by V plus 1 by U. So substituting the terms, 15, 1 by V, we have to calculate. This is U is minus 10. If I transfer this, 1 by V is equals to 1 by 10 minus 1 by 15. Yeah, is this fine? If I take the LCM 15 and 10, it's 5 3s are 2s are, right? Yes? Okay. So the LCM is 30, right? 5 2s are 10, 10 3s are 30. So here it will be 3 minus, so yeah, 2. So it's 1 by 30, V is equal to 30 centimeter. So what 30 centimeter is representing? It's behind the mirror. And they have not given the height of image. So let's not worry about that. That's all. That's done. Okay. Is that clear? Tell me quickly in the chat. Is that clear? Minus 30. Why? Why it's minus 30? It's not front of the mirror, right? It's behind the mirror. And see here, whenever it's behind the mirror, here you have your pole, and I'm telling the image is formed here, so it's positive x-axis, right? Okay, no worries. Done. Yes? Okay. So moving forward, the second part of the question in the OR format they have asked. So let's solve that. Let's see what they have asked here. Okay? Yes, virtual and erect and positive. Yeah, second part of the question. An optical component and an object, yes, is placed along the optic axis. Okay? The distance between object and component can vary. Okay. So what type of image can be formed in the above? So here there is some object, and what type of image is formed? The properties of image they are asking. So what is the answer? What's the answer for this question? First of all, which mirror is this? Which type of mirror is this? So see here. Which type of mirror? Quickly, tell me the answer. Yes, tell me the answer. What's the right answer? Point sized, virtual, erect. First of all, tell me which mirror is this? Is this convex? It's concave mirror. See here, the light is incident in this direction, right? It is curved inward. So this is the reflecting surface, not the outer. Okay? It's a concave mirror. Okay, now tell the properties of image. Properties, okay, no worries, fine. Now tell me the properties of image. What are the properties of image formed by concave mirror. Yes? Can you guys tell the answer? Yeah, quickly. No worries. Okay, that's fine. That's fine. Yeah, all the conditions are possible because we can move this object, right? It's anywhere, the distance between the object and component can be varied. Okay, can be, so where it is, can be varied, nothing but they can move this object. If this object goes to infinity, you'll get point sized image. If it comes near, diminished, then size will increase, it will get enlarged. So all the properties of image will be present in this condition. It can be virtual, it can be erect, it can be real, inverted, every condition are possible, right? So that's fine, that is absolutely right. Every conditions can be present here. But what about second condition? Which mirror is this? Now tell me, which mirror is this? How is the surface? The second condition, what kind of mirror is that? What type of mirror is formed in the above is nothing but, yeah, in this diagram. This is for this question. Now for this question, you have to tell which mirror is this. It's convex mirror, right? It's a convex mirror. So what type of mirror is formed in the above diagram? It's convex mirror. Now draw the ray diagram to form image of convex mirror when object is placed at focus. So complete the ray diagram they are asking. Yes, convex, virtual, erect. Yes. Diminished, virtual. Yes, absolutely. So here you will be getting a small sized diminished image will be formed, and that is the right answer. Yeah, positive, virtual, erect, it has, yeah, absolutely, right. Okay, this is the question on the ray diagram, okay. In yesterday's class also, I have explained you these ray diagrams, okay, about the lens. Tomorrow I will be dealing. So this question is on the concept of ray diagram. Done with that? Yes. How object can be at focus? In convex mirror, you just see two diagrams, right? You see only two ray diagrams. One is at infinity and, yeah, not particularly at focus. You can consider it around focus, okay? Here it is focus, somewhere near to focus the object is kept. We can consider it like that, right? If CBSE considers this to be a wrong question and if that gives you grace marks, that's good. Yeah? Okay. So, yeah, shall we move to the next question? Okay. So, yeah, it's, yeah, B is equals to, right? So, what we have done? Just a second. I'll just check out. Okay. One second. There's a error. It's minus. Okay, focal length is positive. Sorry students, this is just a symbol I missed here. So no worries, I hope. It's positive. So it is 6. This is 30 by 6 and you can cancel out. So you'll get the answer to be 6 centimeter. Fine? Fine? Okay. So yeah, real image, you can read out these things, right? I have explained this already. Yes, I have explained this already. Moving forward to the next question. Yes, moving forward to the next question. A narrow beam PQ of white light is passing through a glass prism ABC as shown in the diagram. Okay. So what they are asking, trace your answer in the sheet, okay. Write the name and cause of the phenomenon. Which phenomenon is, you can see here. Tell me quickly. Tell me the answer quickly. Which phenomenon you will see here? Yeah, what's the phenomenon that undergoes when light passes through a prism? Tell me quickly the answer. What happens? Yes, what happens to the light? What happens? Dispersion of light. Yeah, absolutely. We'll see the phenomenon of dispersion of light. Yeah, dispersion of light. And then what they have asked? Where else in the nature you'll see this phenomenon? In the nature, where do you see this phenomenon? Rainbow. Yeah, dispersion of light through a rectangular prism. It bends the light into 7 colors. Absolutely right answer. Okay. And what is the phenomenon that you see in the nature? That is rainbow formation. Yes, the beautiful rainbow on the sky is an example for, yeah, about rainbow formation. In the last session also, during solving the sample paper, I have discussed about that. Okay. Okay then, moving forward, based on this observation, state the conclusion which can be drawn about the constituents of white light. Yes, now tell me, what happens to the light? It's going to split into 7 colors. From that observation, what you can get the observation out of that? By the seeing the splitting of 7 colors, what answers you can draw from it? You're getting my point about what I am talking. Vib-Gayar series will be formed. Why violet is? Where will be the violet forms? It will bend the least or most? Violet bends least or most? Tell me the answer. Quickly tell me the answer. Prism is triangular. Okay. Yeah. Tell me quickly. Violet bends the least or violet bends the most? Vib-Gayar, where violet has lowest wavelength and red has highest. Formation of Vib-Gayar, we can observe, yeah, wavelength. Yeah, yes, you are telling right about the wavelength. But I am asking about the deviation. Which bends the most and which bends the least? Violet bends the most. It's the right answer. Why? When we see this Vib-Gayar, this violet will be formed here, etc. colors, we will be having red. So red will bend the least and violet will bend the most. Yes, absolutely. Okay, so see here. Why does violet bends the most? It's because of its wavelength, because of the refraction. Okay, violet color bends the most and red color bends the least. So you can check out the answer. Okay, so one property, what you will say? You will say that the white light gets split into 7 colors. That's violet, indigo, blue, green, yellow and red, orange and red, right? Violet suffers the most maximum deviation and red suffers the minimum deviation. Right? Clear with this question? Yes, tell me quickly. Clear with this question? Any doubts? Any question? Yes, students, you're watching the video. Tell the answers in the chat section. Okay. Just type down the answer. Yes, moving to the next question. Wavelength is inversely proportional to deviation. Yeah, violet deviates most. So the wavelength of violet is least. Yeah. Yeah, you can say it like that. Right? So going to the next question. With the help of a labeled circuit diagram, illustrate the patterns of field lines of a magnetic field around current carrying straight long conductor, okay, conducting wire. How is right hand thumb rule useful? So they are asking us about right hand thumb rule. Find the direction of magnetic field associated with the current. Okay. So now tell me, what is the direction of magnetic field? I'll consider a current carrying straight conductor, okay, in which current is flowing in upward direction. Let's consider what's the direction of magnetic field lines. Magnetic field, concentric circles will be formed. Yes, around this we can see concentric circles. They are nothing but magnetic field lines, right? And what right hand thumb rule states? Now they are forming concentric circles, but it is out of the plane or inside the plane. How to tell that? How we can state the direction of magnetic field lines? Put the thumb in the direction of current and the rest of the finger will give the direction of magnetic field. So if I hold this conductor, what's the direction? It is in this direction, right? Yes, yes, yes. We hold the wire in right hand such that thumb points the direction of current. Rest of the fingers will be indicating the direction of magnetic field. Absolutely right. Yes. Yes, Rizal, Krishu, same. Yeah, it's right. Done. Done with this question then? Yes. Now tell me, if I reverse this current direction, what happens? Instead of this direction, if now I'll change the direction to downward, what happens to magnetic field? Tell me that quickly. What happens to the direction of magnetic field line? I have reversed the current direction. It's now flowing downward. Yes, what's the right answer? The field direction is also get reversed. Instead of flowing this direction, I'll keep my thumb this way. So it will be in the reverse direction. Absolutely right. Change will be observed. Yes, 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 that's right. Okay. Is this clear? Fine? Yeah. So here you can see the diagram, right? In the direction field also changes. Yes. Okay. So this kind of question you'll be getting in your examination, right? In the last section of your question paper, you'll be having this kind of passage question. Okay. What I'll recommend you, don't sit and read this passage. We know that battery or a cell. Don't waste time here. You'll get a passage, move forward, check with the questions. See the question, write down the answer. If you feel that I'm unable to answer this question, then only go back to that paragraph, okay? Yeah, it's too long question, right? So just leave it because the question is actually here. Okay, you know the best trick of this? When you'll get this passage questions, right? It may be any subject, I guess. It may be physics, chemistry or bio. Particularly, I know for physics. So rest of the things you can try out there also. Instead of reading that long passage, you can directly jump on the questions. So see here. Explain Joule's law of heating. Do you want to see that passage? Without seeing that passage, you can state Joule's law of heating, right? So what is Joule's law of heating? Tell me the formula. What's the formula or relation that states Joule's law of heating? Quickly. Just tell me that. Yes, Joule's law of heating. H is equal to I square RT, right? So you have to not read that passage. There is no need of reading the passage, right? We know already H is equal to I square RT. That state Joule's law. Explain Joule's law they have asked. So you'll write the same thing. Yes, yes. H is equal to I square RT, right? So you have to state this formula in the definition. What you'll write? Heat produced in the conductor is directly proportional to square of current flowing through the conductor. It's directly proportional to resistance. It's directly proportional to time of flow of current. That's all. First part of the question is done. Right? First part of the question is done. Okay. Now what about the second part? Just a second. Just a second, I'll check out this. Yeah. Can you answer the second part? Yes? Yeah. What's the second part? In practical situation, when electrical appliances is connected to unknown voltage source, then how does the heating effect of electric current can be calculated? No need of thinking again. Second part is confusing. Okay, let me deal with that. How Joule's law of heating is derived? So, is there derivation of Joule's law of heating? We will be discussing about that in detail during electricity chapter. Okay. So, they are asking us explain nothing but the statement and the step you can write, the formula you can write. Okay. Let's deal with the second part. So, yeah. In a practical situation, when electrical appliances is connected to unknown voltage, nothing but you don't know the voltage. The value of voltage is not given and you have to calculate heat generated in the conductor. Directly, simply you'll use this formula, that's all. That's all for this question. Okay. That's all for this part of the question, that is you don't know voltage and you want heat. And what is next? Write the relationship between heat energy produced in the conductor when potential difference applied across its terminal and current flows through the time. In this condition, you don't know what is R. Now, what will be the formula? What will be the formula? Yes, it's an easy question. So, now tell me, what's the formula here for the second OR condition? What will be the formula? Again, we'll go to Ohm's law. That's V is equal to IR. Instead of R, I can write V by I. So, what's the formula for the second question? H is equal to VLT. So, see here. I square in the place of R, I will substitute V by I. And here I have T. This I cancels. So, remaining I will be having I into V into T. So, this is the answer for second question. Yes, absolutely right. It's H is equal to VIT or IVT. Done? So, done with these questions? Tell me quickly. Did you guys understood today's session? Did you guys enjoy today's session? Tell me quickly. Did you guys understood the problem solutions? Yes, tell me. Okay, we have another question. No, it's the answer. So, yeah, we have dealt with this, right? Yes? Okay. So, tell me, understood? Yes, done? Understood each and every question. So, yeah, every Sunday we'll be solving this, right? Every Sunday we'll be doing marathon session in which we'll be solving sample papers, physics, chemistry, bio. Sorry. Okay. So, your science paper will be, sample paper will be discussing here. So, don't miss out the, yes, easy session of the day. Oh, was it too easy? Let's bring some difficult questions next session, right? Let's bring some difficult questions from the next sessions. No worries. Let's start with the easy and let's cater to difficult ones. Okay? No problem. So, yeah, who haven't watched the session, that is sample paper 1, which has been done in the morning, you can go and check out that video also. Okay, yeah. Okay, sign convention. Sign convention, you can go and check out the video which I have done, right? In that I have explained 2 times. Okay, the sign convention of lens you want. Lens is also very similar to mirror. Yesterday I have explained about mirror, right? Yes, bye Gagan. Yeah. So yesterday I have explained you, right? So no worries. Again I'll tell you. Just consider your mathematical graph. The origin, okay, origin in the maths will be pole in the mirror. Okay, will be pole in the mirror and optic center in the lens. Okay. Yeah, I'll tell you. You have the exam. No worries. I'll tell you here only. Okay. So this is x-axis. We call it as positive x-axis, right? So any ray or any distance you are measuring towards right will be considered as positive. Okay. Any distance towards left is negative. If the image is formed this type, okay, means it is moving upward, right? So if it is moving upward, here it's positive y-axis, and if the image is downward, it is negative. So this is the way you have to take. Yeah, about the lens only I am telling. Lens is also nothing different. It is very similar. Instead of pole, we will be having optic center. Okay. Instead of center of curvature, we will be writing 2f. And focus you will be having same. That's all. No big change in the lens. Okay. It's very similar. Most difficult question. Okay. The bill question you want. Okay. I'll be discussing about that. Calculating the cost of electricity, right? Yes. Kilowatt hour. Okay. Yeah, we will be discussing that questions also. Don't worry. So, is the sign convention clear for you? Yes. Is the sign convention clear? Yes. Okay. Done then. Okay. Okay. We will be discussing about the questions also. We will be covering the session also. Tomorrow, don't miss the second part of your light chapter. That is about refraction I will be dealing. Okay. Yeah. Done. Very good. Very good. Very good. So, yeah. We have completed our physics questions too. Yeah. Done. Yes. Let's bring some more difficult questions so it will be more interesting. Right. It will be more fun. So, yeah. Okay. Done. So, done. We are done with today's session. We have solved sample paper 1. Sample paper 2 is done, completed now. So those students who didn't check out the videos, you can go and check it out in the channel. Yes. Yeah, any doubts, any questions? So yeah, anything if you have the things you can ask. So subscribe the channel if you haven't subscribed yet. Subscribe the channel. If you like the videos, hit the like button. Okay. We'll be bringing strategy sessions also. So stay tuned. Yes, stay tuned to the channel. Is that done? Can I sign off? Is everything done, clear? Tell me quickly, done? Yes. Yes. I'm waiting. Tell me, is the session done? I hope. Yes. Okay, fantastic. So, yeah, we have completed today's session. Yeah. Okay. So, yeah, students, thank you so much for watching this video. As I said, if you like this video, just hit that like button and share the videos with your friends too. Let them also join and let's learn together. Okay. So, yeah, thank you so much for watching this video. Thanks a lot. Bye-bye. Let's meet in the next session. Thank you.